So in this lecture, we're going to finish up our discussion of how we go from DNA to RNA to proteins. So this is our central dogma, and today specifically we're going to focus on how we go from proteins to phenotype. And if those words don't make sense to you, on the next couple of slides I have some refresher for you guys. So here's a reminder of our central dogma. So we go from DNA to RNA to protein. So that's the major idea here. However, we also go from information, which is encoded in our DNA, and we go to functions. So through the processes of transcription translation, we go from DNA to proteins, and those proteins contain certain functions, or they do certain functions. Another way you can think of this is that DNA is our genotype. So remember, genotype is our sequence of DNA. And through transcription and translation again, we go to proteins. So proteins, in part, determine our phenotype. So our phenotype is our displayed traits. So again, this idea of genotype, that's our DNA sequence. So the genes that determine your hair color, or the sequence of those genes, is your genotype, and your phenotype is the displayed trait, so in this case, your hair color. So in today's lecture, we're really just going to focus on how we go from proteins to traits. So that is, how proteins determine our phenotypes. And essentially the major idea here is that mutant proteins, so proteins that have some sort of variation that we don't consider normal, so it's not average, or that's not common to the population. So we call those mutant proteins, or variants. So really, there's not actually something considered a mutant protein. So just be aware of that. We're essentially just comparing to what's normal or average. So these mutant proteins can oftentimes cause disease. And so these mutant proteins don't just show up from the middle of nowhere. So remember that your genotype determines your phenotype. Probably better said, we go from DNA to RNA to protein. So if there's a mutation or change in the nucleotide sequence of your DNA, that's going to change the protein that's made. And again, if you have a mutant protein, that can cause disease. So for example, some people have mutations in a growth hormone, a growth factor. So it's actually a protein. So people that have mutations in that growth factor, so they have mutations in their DNA, that mutation in their DNA is then manifested into the protein. So that protein is not working, and as a result of that, we get the phenotype of dwarfism. So really, to discuss how proteins influence phenotype, we need to talk about enzymes. So we discussed enzymes in chapter 2 very briefly. So these are proteins that actually speed up reactions, or for some reactions, even allow them to occur at all. So some reactions are so slow or unfavorable, so they're not likely to happen, that they need an enzyme, so a special type of protein, to drive that reaction. So this is a rather simplistic definition. If you take more advanced chemistry or biochemistry, you'll learn more about how enzymes specifically work and a more proper definition. But for now, know that they speed up reactions. So a key word you may hear is that enzymes are catalysts. So catalysts are things that speed up reactions. However, catalysts are not always enzymes. So catalysts are not always necessarily going to be proteins. So they can be things like platinum, zinc, sometimes even silver itself can be used as a catalyst. So enzymes are always catalysts, but catalysts are not always enzymes. Enzymes, however, though, at least 99% of the time, are going to be proteins. You guys learned about the ribosome. So the ribosome is one example of an enzyme that's actually made up of RNA and protein. So the RNA actually is able to catalyze reactions. So that's an example of an enzyme that's not a protein. But 99% of the time, and in all the examples I'm going to show you in this PowerPoint lecture, those enzymes are going to be proteins. And going way back to chapter 2, remember that proteins have 3D shapes. And those 3D shapes confer special functions. So we're going to see exactly in the next slide how that 3D shape plays a role in how enzymes and proteins function. Before we go there, though, some quick nomenclature. So enzymes usually always end in this ase suffix. So for example, in the bottom of this PowerPoint slide, we're looking at the enzyme catalase. So you can see there's an ase ending at the end. And here we're looking at a chemical equation. So the right arrow or the right pointing arrow means there's a chemical reaction happening. So we're going from hydrogen peroxide. You're probably familiar with putting that on wounds. So if you get a cut or a scrape, you put some hydrogen peroxide on it and it bubbles. So the hydrogen peroxide is broken down into water and oxygen. So water is H2O and our oxygen is O2. So this breaking down or decomposition of hydrogen peroxide normally wouldn't happen too much if you just let it sit out in the open. So here really, this enzyme called catalase is driving this reaction. So the catalase that's actually found in your white blood cells causes this bubbling, which is why when you stick it on a wound, you'll see bubbling. So that's the oxygen floating up and creating those bubbles. So again, this reaction normally wouldn't occur unless you have an enzyme there. So without that enzyme, it's going to take a long time for this hydrogen peroxide to break down. So here's a really nice picture from your book that I think does a good job at showing you how enzymes work. So first we're going to look at the image on the right, showing sort of a simple cartoon version of what's going on the left. So remember, enzymes speed up reactions. So here we have a reaction happening between two of these molecules right here in the red. So the reactants, the molecules that the enzyme is actually going to be working on, is called the substrate. So the more proper term here is substrate, but you can think of them being the reactants. These substrate molecules, you can see there are two of them, then bind to the active site of the enzyme. So remember, enzymes have a very special 3D shape that confers some sort of function. Here, this enzyme has a very nice pocket, or active site, where these two red molecules combined. So you can think of the active site being where the substrate binds and where the reaction is actually going to take place. In this case, we're actually going to see the two red molecules come together. And once that's done, the product pops out and it's ready to go and do whatever it's supposed to do. Notice at the very end of this, that green enzyme remains unchanged. So a defining characteristic of a catalyst is at the beginning and the end of the reaction, they're exactly the same. So this enzyme here can continue to do the reaction as long as it needs to. So the enzyme remains unchanged by this whole entire process. So on the left, we actually have a 3D modeled image of this. So you can see we have that green enzyme again. It has that active site. And you can see those red molecules coming in there and binding to a little pocket where the reaction is going to take place. So this cartoon on the right actually does a very good job at showing you what an enzyme looks like while it's working. But it's important to know that this is literally what's happening. So you have a molecule coming in, binding to sort of a pocket or active site, and then once that happens, you have a change in the protein shape, and the protein starts doing its job. So we've gone over examples of enzymes that break things down. So in the previous example, we had catalase, which breaks down hydrogen peroxide into water and oxygen. Here we're seeing an enzyme build things up. So it's important to note that enzymes can have many more functions beyond this, but the unifying characteristics, they speed up reactions. So for a lot of these examples, we're going to focus on metabolism. And metabolism is actually a very good subject to study in regards to how proteins influence phenotype, because we know a lot about how metabolism happens. So we know a lot of the key players, a lot of the key enzymes, and we also know certain mutations in the DNA that will cause certain phenotypes. So again, enzymes speed up reactions. So for a lot of metabolism where you're breaking things down, if you're breaking down sugars, proteins, lipids, nucleotides, all of those are going to be enzyme-driven reactions. So if you just let a piece of meat sit out in the sun, it's never going to break, or it may take years and years and years of just sitting in the sun and breaking down over time. So what happens though, is that you need to eat the meat. So those digestive enzymes in your stomach start breaking those proteins and lipids, carbohydrates that are in there. So again, these reactions would never happen, or they would happen very slowly without enzymes. So we need enzymes to help metabolize food. In this example, we're actually looking at the metabolism of proteins. Specifically, we're focusing on one amino acid, So remember, amino acids make up our proteins. So we're looking at the amino acid phenylalanine. So we take in some protein that gets metabolized by proteases. So proteases break down proteins. So you find those in your stomach and small intestine. So those break down proteins, and eventually they'll break it down into smaller chunks where we actually have single amino acids, so a single phenylalanine. That phenylalanine that can get shuffled into tyrosine. So tyrosine is another amino acid. From there, we have a lot of other places we can go. We can make dopa. So this is actually a precursor to a neurotransmitter, so very important for our nervous system. Or we can go to thyroxine. So thyroxine is actually a thyroid hormone. So if you've ever heard of your thyroid gland, it sort of sits on the top of your neck. So that thyroid gland produces this thyroid hormone, thyroxine. And it makes that thyroxine based off tyrosine. Before I go any further, I should mention that this is a very abridged metabolic pathway. So there should be many more steps in between each of these arrows. So it takes many, many steps to get from tyrosine to thyroxine. So really the point of this slide is to show you various diseases that affect this metabolic pathway. So for example, the disease PKU, which we're going to talk about on the next slide, is a disease that affects the conversion of phenylalanine into tyrosine. As you're going to see, that can have huge issues in regards to health. Similarly, albinism affects the conversion of dopa into melanin. So melanin is a protein that gives our skin pigment, so it gives our skin color. So that's why albinos have very, very pale skin. And you can see the various other diseases that affects enzymes. So remember, enzymes are catalyzing all its reactions. So whenever you see a yellow arrow, you can then imagine there's an enzyme there. So these various diseases are caused by mutations in the DNA. There's mutations in the DNA that affect the proteins that are made in this metabolic pathway right here. So if that protein isn't working, the metabolic pathway is not going to proceed. So of particular note is the disease PKU. So PKU is actually a very interesting disease because it's one of the very few that we actually test in all of our newborns. So probably just about any newborn that's born in this year is going to receive a PKU test. So PKU is essentially a disease that prevents the breakdown of phenylalanine, or conversion, I should say, into tyrosine. PKU itself can cause intellectual disability if it's not addressed. And of course, the keyword here is addressed. And this is done by ensuring that the baby is fed a low-protein diet. So this is why it's imperative that PKU is tested in young children, since if you start feeding them proteins, as we saw in the last slide, those proteins are going to be broken down. You're going to have phenylalanine, and as that phenylalanine starts building up and being converted into other things, in this case, this phenylpyruvic acid and more, as you can see there are arrows pointing to the right, as it starts getting broken down into those products instead of tyrosine, that will start affecting the nervous system and eventually cause intellectual disability. And the common way to test for this, as you can see, is we have the foot of the newborn. And what they've done is take a little pinprick, and they're going to draw a little bit of blood, and they're going to create a blood spot. Just a tiny bit of blood is what they're going to be using to diagnose whether or not this baby has PKU. And here we have actually another example of how phenotype is driven by both the genotype and the environment. So if you're able to feed the baby a low-fat or low-protein diet, you can essentially alter the phenotype of this baby. So the baby is not going to have intellectual disabilities if you feed him a low-protein diet. So despite his or her genetics, if you alter the environment of the baby, you can prevent the phenotype from occurring. So here are some examples of other phenylalanine pathway diseases. So I won't really go over this, but it's important to be aware of. Next, we're going to talk about diseases that affect something called glycogen. So glycogen is a polysaccharide. So remember, a monosaccharide is a single sugar molecule. So a polysaccharide is many sugar molecules linked together. So poly means many, saccharide refers to sugar molecules. So here we actually have a picture of glycogen. So you can see it has all these little branching chains and it has these little dots everywhere. So each of those red and black dots is a sugar molecule, so one molecule of glucose. In the center, we actually have a protein. So we're actually going to see some diseases that affect glycogen breakdown. So glycogen is how your body stores glucose. So how your body stores carbohydrates. So if your body is unable to break down glycogen, essentially it's going to store a bunch of sugar and not be able to break it down again for use. So glycogen in this polysaccharide form is not usable by the body. It has to be broken down into smaller molecules. And so there are a variety of diseases that can affect glycogen breakdown. So as you can see, we have 4 listed here. And the specific metabolic defect, so essentially what protein has an issue. So for example, for type 1 or glycogen storage disease, there's an issue with glucose 6 phosphatase. So it appears that we're deficient in that enzyme there. So remember, all enzymes end in the ase suffix. So here glucose 6 phosphatase is missing or deficient. Here we can see how it's inherited. So in the next part of the course, we'll learn exactly what autosomal recessive means, but here we're just looking at the type of inheritance of the specific trait. So with a specific metabolic enzyme mutation comes a specific phenotype. So here you can see some of them. So for example, that type 1 glycogen breakdown disease causes severe enlargement of the liver. And lastly on this table, we have the MIM number. So the MIM number actually refers to this website called OMIM. So this is the Online Mendelian Inheritance in Man. So essentially, as it says, it's an online catalog of human genes and genetic disorders. So this is a very helpful database, especially if you're doing the disease project, to look over. So this provides a lot of detailed information, more so than you would get from a textbook or Wikipedia or anything like that, since it actually uses primary literature. So it actually uses scientific papers as its content. So it allows you to learn about a lot of genetic diseases and how genetics affects certain diseases that we normally don't think of as a genetic disease. So for example, I looked up diabetes type 2. So diabetes type 2 is not normally what I would say a genetic disease. So definitely there's a genetic component to it, but there's definitely also an environmental component. When somebody says genetic disease, I think of something like PKU or albinism. Diabetes type 2 is probably not the first thing I would think about. But here, OMIM actually puts together a lot of references and citations that tell you about how genetics actually affects type 2 diabetes. So for example, it has something like this. So as you can see, it gives you a lot of information about what genes may possibly be causing type 2 diabetes. So with that said, we're going to move on to the next metabolic disease. So here we have galactosemia. So galactosemia is essentially the inability to properly break down galactose. So galactose is a very common component of lactose. So lactose you may know from milk. So milk has lots of lactose. You may also know if people are lactose intolerant, so they're unable to break down lactose. But here we're specifically looking at galactose. So lactose is a disaccharide. So remember, di means two. So lactose has 2 sugar molecules, 1 glucose and 1 galactose. In galactosemia, you have an issue with the third step of breaking down galactose. So essentially you have issues converting galactose 1-phosphate into UDP galactose. Everything else though happens normally. So glucose is shuttled off and gets broken down. So really it's the galactose part of lactose, so the second half, that we have an issue with. Lactose intolerance though, will be more here. So lactose intolerance is more about the inability to break down lactose at all. So we can't even get glucose from lactose in people with lactose intolerance. So that affects step 1. But here we're going to be looking at step 3 in galactosemia. Just like PKU, a buildup of galactose 1P or 1-phosphate can cause intellectual disability. On top of that, it can also cause blindness as well. As we're going to see in the next slide, there are variants of the enzyme that breaks down galactose 1-phosphate. One such variant is Duarte galactosemia, or DG. So it's a less severe form of galactosemia compared to other forms that we're going to see in the table on the next slide. So here we're looking at the multiple alleles of galactosemia. So remember, alleles are different versions of genes. So here we have the G plus allele. You can think of this being the normal allele. You have the G superscript D. This is your Duarte. Galactosemia. And then we have a lowercase g. So this means that protein is absent or it's not working. So notice that we have 2 alleles for all of these genotypes. So remember, we have 46 chromosomes, 23 from dad and 23 from mother. So for every gene we have, in this case, the gene to convert galactose 1-phosphate into UDP galactose, we have 2 versions of that gene. So we have 2 versions of the gene, so we have 2 versions of the protein. So for example, this first row right here, we have 2 normal versions of this protein called GALT. So GALT is actually the enzyme that's doing this process right here. So again, for the G+, we have 2 alleles, so 2 versions, one from your dad, one from your mother, and both these are normal, so they produce the normal GALT enzyme. For the G+/G superscript D, we have 1 Duarte galactosemia or 1 Duarte GALT gene and 1 normal GALT gene. So even though we have 1 diseased allele here, we still have 75% enzyme activity. So even though this Duarte allele is causing issues, we still have most of the enzymes working there. As a result, the phenotype is actually normal. And as you can see, as we play with which alleles we have, so for example, if you have the normal allele and this lowercase allele, you have 50% activity. So that still produces a normal phenotype. And in the last row, you can see we have 2 lowercase g's, so essentially this gene is absent, or it produces a protein that doesn't work at all. We have 0% enzyme activity, so GALT is not working at all, and we have galactosemia. So even though this is a very small table, I think it points out a very important fact. So we have multiple variants causing a disease. It's the same exact idea for dwarfism, same idea for albinism. So you can have multiple different types of alleles causing various types or various severity of diseases. So don't think it's just all or none thing. You can have various variants of diseases, and this can be caused by different alleles you may have. The second important note or idea you can gather from this table is that really even if you have 50% of the protein there or 50% of the enzyme, here we're still able to have a normal phenotype. In later lectures, this is going to be very important for understanding how dominant and recessive phenotypes come about. So again, I think even though this table is very small and innocuous, I think this is an important table to understand how enzymes work, and also is going to be important for later lectures when we talk about the transmission of genes. So we're going to shift gears now, so we're moving away from metabolic diseases and focusing on transport protein diseases or issues. And here we're specifically going to focus on hemoglobin. So hemoglobin is a very important molecule or protein for transporting around oxygen in our blood. So hemoglobin is actually a very important protein because it was the first example of a protein with an inherited structural defect. So before that, we really didn't know how we got from proteins to phenotype. So really it was hemoglobin that helped pioneer how we understand how proteins affect us. More specifically, this disease, sickle cell anemia, which we talked about in the first lecture of this course, was important. It provided the first evidence that DNA mutations cause amino acid changes. So this was before our understanding of the central dogma. It also provided first evidence that a single nucleotide change can cause disease. So the change in one base pair in DNA can cause sickle cell anemia. So here's a picture of hemoglobin. So it's a major component of our red blood cells. As you may know, red blood cells are important for transporting oxygen. So the molecule that does that is our hemoglobin proteins. So those are transporting oxygen. So hemoglobin is made up of 4 major parts. So there's an alpha chain 1, beta chain 1, beta chain 2, and alpha chain 2. So it has 4 subunits. So subunits is the technical term here. So 4 parts or 4 subunits. Within each of those subunits, we have a heme, It's not very important for our discussion of genetics, but I thought I would just point it out here. So even though hemoglobin was very simple in the last picture I showed you, it's actually more complex at the genetic level. So it actually turns out that we have different variants of hemoglobin. So we have different hemoglobin when we're an infant than when we're an adult. So the hemoglobin we were looking at was made up of alphas and beta chains. As we're going to see, when you're an infant, you actually have different hemoglobin subunits or different chains making up that hemoglobin. So for example, in an infant, you actually have gamma G and gamma A subunits along with your alpha globin. And you can see there are many other subunits, so like zeta, epsilon, and delta. We also have non-functioning genes. So these are pseudo-zeta, pseudo-alpha 2, and so on. So these genes will then go on to produce various proteins that make up hemoglobin. So really, this gives our body some opportunity to mix and match how we make our hemoglobin. And in the slide I'm going to show you next, you can see a graph that shows you an example of how hemoglobin types switch throughout our lifespan. So before birth, essentially the middle of this graph, we mostly have gamma G and gamma A globins along with alpha globin. So this makes up our hemoglobin for us when we're infants and in our mother's womb. At birth, though, you can see that the amounts of gamma G and gamma A severely drop within a couple of months of being born. Instead, this was replaced by beta globin along with our alpha globin to make our common hemoglobin that you're familiar with and that I showed you the nice picture of. So beta globin, as you're going to see, is actually going to be very important for a disease called sickle cell anemia. So something I brought up multiple times, but sickle cell anemia is actually a genetic defect, specifically in the beta globin gene. So alpha globin is unaffected, so it's only beta globin that has this mutation. So with that in mind, you can think about this graph in a new light. So according to this, it would not be possible for a baby to have sickle cell anemia, at least not before 2 months of age. Since that baby is not producing beta globin, it's not producing that subunit that's going to have sickle cell anemia. And you can start taking this further and thinking about certain diseases that only affect people of certain ages. So perhaps certain diseases have certain protein variants that affect when diseases appear. So it's definitely very interesting to think about in regards to phenotype. And again, it's not just a sickle cell allele and a normal allele. You actually have many variants that you can have. And here's a table that shows the various ones. Of course, don't memorize this, but you can see that there are various phenotypes that can arise due to variants in the beta globin chain. Also, it's important to realize that not all of these diseases are detrimental. So something like HBM Boston, that produces reduced O2 affinity. So probably that person is not very healthy. However, somebody with a Bethesda hemoglobin has actually increased O2 affinity. So perhaps this person would be a better athlete than somebody with just a normal hemoglobin molecule. So even though we focus a lot on disease, it's important to remember that some variants can actually be beneficial for us. So here's a very busy diagram showing you the various effects of sickle cell anemia. You can see it has many, many effects. Not surprising since red blood cells are so important for transporting oxygen. The main takeaway message from this, though, is that we can have one genotype. So here we just have the sickle cell allele causing many phenotypes. Just like those words dominant and recessive, which I mentioned earlier, this idea of one genotype causing multiple phenotypes. So weakness and fatigue, poor physical development, pneumonia, etc. You can see this one genotype causes multiple phenotypes that are unrelated. So this idea will be something we come back to in later lectures. So now we're going to focus a little bit more on sickle cell anemia, so looking at that beta globin sickle cell mutation. So remember the alpha chain, so we have 2 alpha subunits, 2 beta subunits. So in sickle cell anemia, only the beta subunits are affected. So essentially what happens here is this beta globin gene mutation causes hemoglobin molecules to stick together. And the next slide actually have a very nice picture showing exactly what this looks like. Essentially they stick together, they start to distort and get really ugly, and essentially they create something that looks like a sickle. So on the left we have a nice red blood cell, it's biconcave, so there's a little depression in the center, and you can see on the right we have a sickle cell red blood cell, so you can see it's very, very distorted compared to the normal red blood cell. As you guys saw on the last slide, sickle cells can cause a lot of phenotypes. But in particular, it causes anemia, so inability to transport oxygen, pain, tissue damage, heart attacks, and strokes. As you can see, it's a very dangerous disease. So here we're actually looking at the protein level, so we're actually looking at those hemoglobin proteins forming those fibers. So as you can see, it's coming together and getting bigger and bigger. And as those fibers form, you can see it creates something like this. So it creates that characteristic sickle cell shape. So really the scary thing about sickle cell anemia is that it's caused by a single base pair change. So in the normal hemoglobin, as you can see in the green, we have CTC. In the hemoglobin sickle cell anemia allele, we have CAC. This of course gets transcribed and translated. In the normal protein, we have glutamic acid, and in the hemoglobin sickle cell anemia, we have valine. So single base pair change causes a single amino acid change, and this causes a very devastating disease. So very, very frightening to think that it could be this simple. So with that said, we're going to move to a more lighthearted example, and this is the chemical phenylthiocarbamide, or PTC for short. So PTC is actually a molecule that's found in kale, cabbage, Brussels sprouts, and broccoli. So one thing you may find in common between all these vegetables is that certain people seem to really dislike them. And it might be because this PTC causes bitter taste. So again, PTC is a chemical found in all these vegetables. However, certain people cannot taste PTC. So people who cannot taste PTC won't get that bitter taste. So you can imagine they'll like kale, cabbage, Brussels sprouts, and broccoli a lot more than people who can taste PTC. So of course you can start thinking about obesity and how PTC tasting may affect that. Of course, this is only one example, and there are many other genes that affect what we eat. So again, I said that PTC is not something everybody can taste. And here's actually a very good example of population genetics. So here we're tracking the non-taster allele. So essentially people's inability to taste PTC. And as you can see, certain populations have this allele more frequently than others. So for example, Australia and parts of Alaska have a lot of people who can't taste PTC. And of course, then you have to go wonder, does cabbage and broccoli sell more in Australia and Alaska than it does in other parts of the world? So people's inability to taste PTC is actually driven by protein. So just like we saw in all the other examples, mutations in genes cause changes in proteins, which change our phenotype. In this case, our phenotype is the inability to taste PTC. So here we have an example of a taste bud, essentially. So you can see the taste bud is made of multiple cells. So here we have taste receptor cells. And those are connected to nerves that run to our brain. So what happens is that food comes along, it enters into this taste pore, and then binds to cell surface receptors. So the food particles don't actually enter into the cell. So you just bind to the receptors on the plasma membrane. So remember that plasma membrane we talked about in chapter 2, that's made of both lipids and proteins. So there are actually proteins sitting on there that are able to receive signals from the outside environment. In this case, able to receive the food particles and convert that into nervous impulses. So essentially, it receives the food that binds to a receptor, and those signals are then sent to your brain, which processes the information. So people who can't taste PTC actually have a variant of this cell surface receptor that prevents them from tasting PTC. So there's some change in that protein that prevents PTC from binding or prevents PTC from activating it, which is why they can't taste it. This is very similar to certain olfactory abilities. So like taste, olfaction or smelling is driven by cell surface receptors. So we breathe in particles, and certain cells in our nose have certain cell receptors that those particles bind to. Those cells then convert that signal into electrical impulses that our brain understands as smell. So an interesting study pointed out that 2/3 of people tested could only smell pink flowers but not red ones. The rest of the people there could smell red flowers but not pink ones. Lastly, we're going to finish this exercise with some clinically relevant examples, specifically focusing on prescription drugs and genetics. So genetic variations can actually determine our sensitivity to drugs and the presence of side effects. So for example, here we have this drug, tamoxifen. So tamoxifen is actually a drug used for breast cancer. Tamoxifen by itself, though, if you just take the pill, is inactive, so it won't do anything to your body. It's because of a liver enzyme. So CYP2D6 or cytochrome P2D6, that tamoxifen gets changed into endoxifen. So again, enzymes catalyze reactions. Here we're catalyzing reaction from tamoxifen to endoxifen. Endoxifen, unlike tamoxifen, is active, so it actually do something. In this case, affect estrogen in the female body. So as you can imagine, if there are mutations in the gene that codes for cytochrome P2D6, this can affect the sensitivity of a person to this drug. So for example, if they have lower levels of CYP2D6, or they have an inactive form, you may have to consider a different treatment or increase the dosage of tamoxifen. And this idea of changing doses related to genetics and understanding how genetics plays a role in pharmacology is pharmacogenetics. So it's a study of genetic variations that underlie the differences in individual responses to drugs. So as I gave you an example, variants in the CYP2D6 enzyme can cause various responses to tamoxifen. Another good example is this drug warfarin. It's a common blood thinner, so it prevents blood clots, so it prevents strokes or heart attacks. And it's actually a very common drug that's prescribed. So just like tamoxifen is broken down by cytochromes, so proteins in the liver, here warfarin is broken down by CYP2C9. So in the graph below, we have very common dosages. So they actually took a survey of doctors to see what dosages of warfarin they're prescribing. So with somebody with a normal CYP2C9, about 30 milligrams or 35 milligrams was common per week. For somebody with these variants, about 25 milligrams per week. Somebody with these variants, 20 milligrams per week. And these variants at the very end, those about 10 milligrams per week. And you can see the error bar is very wide, so almost 1 or 2 milligrams all the way up to 20 milligrams for those last variants. So essentially, you can see how the variation in the CYP2C9 cytochrome or liver protein affects how much warfarin they prescribe or the dosage they prescribe. It's very important that you're aware of this as a clinician, because if you prescribe too much warfarin, if they get a cut or something happens to them, this can cause life-threatening bleeding. So blood thinners prevent clots, and they also prevent clotting of wounds. So if you get a cut and you're on a very high dosage of warfarin, you could very possibly bleed out from that single cut.